Hi, today I'm going to explain about gene expression. Genetic information flows from DNA to protein involve two steps, which is transcription. Transcription is the transfer of information from DNA to RNA. It occurs in the nucleus. The second step is translation. Translation is the transfer of information from RNA to protein. Transcription occur in the nucleus, involve RNA polymerase enzyme. During the transcription, one DNA strand acts as the template to synthesize a complementary strand of mRNA. Note that transcriptions involve three processes, which is step 1, initiation, step 2, elongation, step 3, termination. Step 1. Initiation. RNA polymerase binds to the promoter of DNA. It moves along the DNA in the direction of transcription. It unwinds the double helix DNA. Next, RNA polymerase breaks the hydrogen bond between the complementary nitrogenous bases. Both the DNA strands are separated, and one of the DNA strands is used as a template. Step 2. Elongation. RNA polymerase add nucleotides in the direction of 3' to 5' end. RNA polymerase add a free ribonucleotide which are complementary to the bases on the DNA template strand. Uracils pairs with adenine, cytosine pairs with guanine. Newly synthesized mRNA detached from DNA template, DNA double helix reform or rewinds, and leaving only few DNA-RNA hybrid region. Step 3. Termination. Elongation will proceed until RNA polymerase reach termination site. Terminator sequence gives signal to RNA polymerase to stop adding nucleotide to the mRNA strand. RNA polymerase will detach. mRNA molecule is released. Double-stranded DNA will rejoin to form double helix again. In prokaryote, mRNA doesn't need any modification, but in eukaryote, further modification is needed. Thus, after transcription, its mRNA is called as pre-mRNA. Next, activation of amino acid. mRNA codon carries the code for amino acids. Anticodon of tRNA has a specific complementary binding sequence for the codon of mRNA. The RNA that binds to the codon in mRNA must carry the amino acid encoded by that codon to the ribosome to ensure accurate translation. The RNA will bind with the amino acid that match the code for the correct mRNA codon. Each kind of tRNA binds to a specific amino acid. Activation is a process in which a specific amino acid is attached to a tRNA molecule to form amino acid tRNA, catalyzed by amino acid tRNA synthetase. ATP is used as energy source. There are two different amino acid tRNA synthetase, and tRNA is reactivated many times. On the other hand, amino acids are linked to their respective tRNA molecules by formation of ester bond at the 3' end of tRNA. Translation. Translation involves three processes, which is step 1, initiation, step 2, elongation, and step 3, termination. Step 1. Small ribosomal subunit binds to mRNA. It binds to the specific nucleotide sequence just upstream of the start codon, AUG. Initiator amino acid tRNA that carries methionine binds to the start codon held by hydrogen bonds. The sequence of start codon 5' AUG 3' is complementary with anticodon 3' UASC 5' found in initiator tRNA. Then, large ribosomal subunit attached with the initiation complex To complete the translation initiation complex, protein called initiation factors are needed to assemble all these components together. Energy is provided by the hydrolysis of GTP. Initiator tRNA is located in the P site of the ribosome. A site is empty, available for the next amino acid tRNA to enter, that has a complementary sequence of anticodon to the second codon on mRNA. In step 2, elongation. In the elongation stage, amino acids are added one by one to the previous amino acids at the C terminus of the growing chain. Each addition involves a 3-step cycle. One is codon recognition, second, peptide bond formation, third, translocation. Energy is supplied by hydrolysis of GTP in step 1 and 3. During codon recognition, amino acid tRNA with anticodon, which is complementary to the mRNA codon, enters the empty A site. Peptide bond is formed between the carboxyl group of amino acid in the P site and amino group of the new amino acid in A site. It's catalyzed by peptidyl transferase. Large subunit, which is a ribosomal RNA, act as enzyme, thus called riboson. These steps remove the growing polypeptide from the tRNA in the P site and binds it to the new amino acids on the tRNA in the A site by a peptide bond. And then ribosome moves a codon ahead from 5' to 3' direction. tRNA in the A site is now translocated to the P site. This is called translocation. Initiator tRNA is now in E site, exits from ribosome. A site is now empty. The third amino acid tRNA enters A site. Peptide bond is formed between second and third amino acids. The growing polypeptide chain is released from tRNA in the P site and bind to the amino acid attached to the tRNA in the A site by a peptide bond. Amino acids are added one by one to the previous amino acid. The whole process is repeated until all the codons of mRNA is translated. Step 3. Termination. Elongation continues until stop codon in the mRNA reach the A site. The stop codon, which is UAG, UAA, UGA, do not code for amino acids, thus no tRNA will bind to the stop codon. Stop codon acts as a signal to stop translation. A release factor is a protein that recognizes the stop codon binds to the stop codon in the A site. Then, the release factor causes the addition of water molecules to the polypeptide chain. This reaction hydrolyzes the bond between the last amino acid of the polypeptide chain and the RNA in the P site. Newly synthesized protein is released, and ribosomal subunits is dissociates.